We're really happy today to be bringing you Dr. Jacqueline Burke. She's a research assistant professor, biomedical engineering, and the Tolerance Lab. She's also the CSO at SNC Therapeutics Incorporated. And the title of her talk is Subcutaneous Nanotherapy for Type 1 Diabetes Onset, Delay, and Prevention. A little bit about her. She is a research assistant professor of biomedical engineering, as I said, at Northwestern University. She received a BS in biomedical engineering from Johns Hopkins in 2017 and an MS PhD in biomedical engineering from Northwestern in 2022. And following her graduate studies, she pursued entrepreneurial ventures alongside research assistant professor appointments, first in pharmacology at the University of Illinois, Chicago, and now in biomedical engineering at Northwestern. She started AbbVie Ventures funded SNC Therapeutics, which is a startup company focused on therapeutic delivery, where she currently serves as the chief scientific officer. She has a recent review paper out in Frontiers, which came out in May of this year. I'm going to drop it in the chat, and feel free to drop questions in the chat as she's talking. She's happy to answer them, and we'll also take questions at the end. Thank you so much, Jacqueline, for joining us. This is a really timely and important topic near and dear to our hearts. We just attended ADA, and there was a lot of buzz around this type of, or new types of delivery and protection of transplanted islets. So we're really interested to hear what you have to say.
Thank you so much for the kind introduction. Yes, feel free to interrupt me if you have any questions. Just say something if I'm not looking at the chat. So
I'll be looking at it, so I'll let you know.
Perfect. Thank you. So like you said, I am interested. Here's one disclosure quickly. In my lab, we focus on biomaterial strategies to overcome autoimmune diseases. And so one of the most exciting strategies right now is polymeric nanocarriers. And so these carriers are the next generation of delivery vehicles for both small molecule therapeutics, RNA, etc. And so here you can see, in the past, we've looked to liposomes and LNPs, lipid nanoparticles, but now we're moving towards this polymeric vehicle. And this is because in addition to being very, very stable, we're able to deliver these polymeric vesicles via subcutaneous intramuscular injection in addition to IV injection. And they target the lymph nodes. And for autoimmune diseases, this is a really critical area where a lot of these immune cells that we want to target are located, and they reach systemic circulation without being cleared that you would see with something like oral administration. And so we're able to target a lot of downstream organs very, very nicely. And the biomaterial that we're going to focus on today is PEG, polyethylene glycol, block polypropylene sulfide. So this is a diblock copolymer. And you'll see, obviously, PEG is hydrophilic, and the polypropylene sulfide is hydrophobic. And this forms these really nice polymerisome nanostructures. And we really like these because by using just one simple polymer component, they're a lot more stable than your LNPs, where you have to choose, percentage of lipids, etc. for every different formulation. You just have something off the shelf. And they're also very scalable in this regard, meaning that we can have a large batch production more easily. We can also customize them as you can with LNPs. Our first kind of proof of concept was showing that these particles were safe. So we've shown in mice and also in non-human primates that these particles given at very high doses, which we probably would never give clinically, are well tolerated. We didn't see any complement activation or anti-PEG antibodies within the primates of note. So why do we kind of want to use these particles specifically for a tolerance application? And one of the interesting things about these polymerisomes is they are highly uptaken by antigen-presenting cells while avoiding T cells. And so this is because particles dictate what cells they're going to be uptaken by what proteins are denatured on the surface of the particle. And so we have these scavenger receptors that denature and are uptaken by these antigen-presenting cells. And the polypropylene sulfide is oxidation sensitive, meaning that once the particles are uptaken, the payload releases. We also avoid T cells. These cells are important, but we are going to target the T cells via the antigen-presenting cells. And I'll show you why that's really, really critical. So we all know here that type 1 diabetes is really hard disease to live with, and it's affecting more and more people every day. This is caused by the autoimmune attack on our insulin-producing beta cells, and insulin, gets us through the day, but it causes long-term side effects due to time out of range. And this has a lot of devastating effects, both for our economy and for patients and families dealing with this disease every day. And so in my lab, I focus on both treating patients with existing type 1 diabetes or looking to treat them in the future via allogeneic islet transplantation, and then also preventing new patients from having type 1 diabetes onset. And so with the islet transplantation, while this is a curative therapy, you are left with a lifetime of immunosuppressive drugs that you have to take. And, as they deem it today, the immunosuppressive therapy side effects are worse than the potential problems of living with diabetes as it is managed today for most typical patients. And then prevention, the drug we have on the market today is Tzield, an anti-CD3 antibody, the only drug on the market that's been available for about 2 years. Because it's an anti-CD3 antibody, it has immunosuppressive transient properties. And so we're looking for something better and something that can delay for longer than 2 years, which is what we get with Tzield. And so one of the drugs we've looked to is rapamycin. So rapamycin is a very hydrophobic drug. It's used as an immunosuppressive therapy for transplantation applications, but it has a variety of adverse effects. And these include like mouth sores. It's also metabolized in the gut, so you don't get all of the drug you take when it's given orally. And it goes everywhere in the body. So you're not just targeting the cells that you need for immunosuppression or tolerance, you're targeting every single cell. So we found that the interesting thing about rapamycin is that it has very specific cell type effects. So when you give it to a different cell population, it has different effects. So the intended target is T cells, and rapamycin will inhibit their proliferation. While that is great for preserving a transplanted organ, it does leave these patients immunocompromised, susceptible to infection, etc. It also has a number of adverse effects when it targets other cells. So in the liver, it can cause lipid dysregulation, which can be seen throughout the body in a variety of different lipid disorders. However, when you give it to antigen-presenting cells like dendritic cells, macrophages, monocyte-like cells, the co-receptor expression is reduced and you have a tolerogenic phenotype. And so we wanted to harp on this and say, okay, can we just give this drug to these antigen-presenting cells while avoiding these other cells to harness this tolerance property? And we wanted to use our specific delivery vehicle that delivers to antigen-presenting cells to do this. And so we developed these tolerisomes. So these are our nanoparticles we talked about earlier, loaded specifically with this rapamycin. And we can give this drug subcutaneously, and it drains to the lymph nodes, which is really critical because that has a lot of the cells that we need for tolerance. And so in a patient that either had an islet transplantation or has type 1 diabetes onset, you have the presentation of islet antigen at the same time as you're giving these particles. And together, the presentation of the antigen with the particle signals to T cells for true tolerance. And this is mediated via upregulation of CD8 positive regulatory T cells. And these are a unique population of T cells. They're actually more potent than CD4 positive regulatory T cells to help with this tolerance induction. And so things that we really like about this therapy, it's non-lymphodepleting. So we're not reducing your T cell populations. You can give it subcutaneously. Tzield, one of the issues is you gotta go to these intravenous infusion centers and have therapy for 2 weeks, etc. This could potentially be done in your home. The polymer and rapamycin have no issues with redosing. Some of the early studies with anti-CD3 had issues with redosing. And this is kind of an antigen agnostic therapy, given that we're relying on the timing of the antigen presentation for tolerance. And in that regard, it's also a personalized therapy. So how does this specifically work? Let's see kind of the immunology. So we have a co-stimulation blockade induced with our tolerosome in dendritic cells, macrophages, and monocyte-like cells. So I'm just showing you the DCs here, but we see reductions in CD40, CD80, and CD86 in these cell populations. And this is paired with a targeted MHC2 increase. And we see this in the same cell populations. And so MHC2 and the CD4 T cells go hand in hand in their signaling, and we see the CD4 positive T cell antigen. And as I said before, we have these regulatory T cells that help restore our autoimmune function, our CD8 positive Tregs, increasing with our tolerosome therapy. So like I said, the tolerosomes, they act on our antigen presenting cells, and they signal to our T cells to induce tolerance and to be accepting of this islet antigen, as opposed to suppressing the T cells like some of the other rapamycin, tocilizumab, etc. So let's show how we did this. So the first experiment we did a islet transplantation using bulb C donors transplanted to B6 recipients. And so this is a very robust immune response model. And when we give our tolerosomes, we see about 83% of the mice remain normoglycemic after 100 days post-transplantation. This is very exciting. This was done in the interportal liver model, which is the most clinically relevant model. This is the standard for clinical practice currently. We also did it in the kidney capsule model. This is a little bit more of an academic model. And the nice thing about the kidney capsule model is that you can graft it out. You can remove the kidney and remove the islets and show that it was indeed the islets that were causing this normoglycemia. So when we take the kidney out, all of the mice become diabetic again. So just proving the model is robust. We also wanted to demonstrate antigen-specific tolerance. So we have our same islet transplantation setup here. And after 100 days of transplant, we do a challenge. So we take the T cells from our recipient mouse, and we challenge with antigen from our islet donor, our bulb C mouse, and a foreign mouse, a C3H mouse. So we want to see if the T cells will proliferate in response to this challenge. So for our donor mouse, we want the T cells to be quiescent, not proliferate, where we still want a robust response to this foreign stimulus. And that's indeed what we see. We see both CD4 and CD8 immune response to the foreign challenge, where we see quiescence in response to the donor mouse. So we next wanted to take this in and say, okay, we can do islet transplantation, but what about a T1D onset delay? And so we utilize the NOD mouse. So this is a non-obese diabetic mouse model. It spontaneously develops diabetes as it ages. So you'll see the beta cell mass decreases while blood glucose increases over time. So about half of the female mice will become diabetic at about 18 weeks of age. It varies a little bit by litter. So we first did a preliminary study, only 6 mice, and we just did half of our dose that we did in the islet transplant studies to see what would happen. And we paired it with a non-treated control. And what we see is in this chart, this is the age of the mouse over here. And then this is the diabetes incidence. So what percentage of the mice develop diabetes? And people like to refer to the age of onset as the age when 50% of the mice are diabetic. So kind of the median incidence age. And we see with the control mice, we have about 50% diabetic at that 18-week point. And with our treatment, we extend this out to 27 weeks. So that's really exciting because that's a 9-week delay. And I'll show you some of the results for tocilizumab or the mouse version of Tzield and what they had as well. So this is great. We also saw that fewer mice actually became diabetic overall, which kind of leads us to hypothesize that maybe prevention could be a possibility as opposed to just the onset delay. We wanted to do the head-to-head with the Tzield, the only drug on the market for onset delay. And on paper, we thought, we had a lot of advantages. Our therapy is non-lymphodepleting. You can give it at home via subcutaneous injection, and you can redose without the risk of anti-drug antibodies. So we did this, and we see here now giving the full 6 doses, that with a control, we had about 24 weeks of age for median onset, and then with Tzield, 27 weeks, and then we extend to 33.3 weeks. So this is very exciting, another 9-week delay, and 6 weeks further than the anti-CD3 mouse Tzield. So very exciting here. We also see some degree of prevention, but this is something that we still need to explore a bit more here. And of note, this was performed at the 12 weeks of age. So we're intervening at 12 weeks of age in these mouse lives. So this is kind of a later stage intervention. And this was the point in the preclinical data package for Tzield that was the best for this particular anti-CD3 antibody. But we also wanted to see how it would be at 8 weeks of age. If we could intervene earlier, that's always great. And we also wanted to understand our dosing a bit better. So in this case, we have our control and Tzield coming in around the same. Again, this isn't the best point to look at for anti-CD3. And we extend to 29 or 32 weeks, depending on the dose that we give. So we have delay and perhaps some prevention, something that we need to explore further. So finally, one of the things that can be a pause for patients is that about 73, 75, depending on what clinical trial you're looking at for tocilizumab, these patients develop lymphopenia, and it can be very severe. It can be grade 3 lymphopenia, which is not great. So it's transient. It's just only during the dosing. But if we could avoid it, maybe that would be something to consider. And so we just show here that regardless of dose, lymphopenia is not something that is an effect with our drugs. And so that might be a helpful safety feature as well, especially if patients are out in the world where they might be vulnerable to infection. So I've shown you that we're able to develop these rapamycin-loaded targeted delivery vehicles to direct rapamycin to antigen-presenting cells while avoiding the T cells. And so we're able to achieve a tolerogenic state without immunosuppressive side effects. Going forward, we are currently working with transplanting human and porcine islets to humanize mice. And we're doing that with Matt Brown at the University of Wisconsin. And we're also doing some non-human primate dosing studies with our tolerosomes. And so this is being done with Dr. Ali Naji and Ching-Rong Liu at Penn. And these are both funded by Breakthrough. So we're very grateful for that and excited to kind of see more data here. And so I'd like to thank my group as well as the funding sources, and happy to take any questions you may have. Very grateful for the invitation. So thank you.
Thank you so much. I had a question, if you think about this, in terms of each extension that you were able to achieve with the NOD mouse delay, have you guys thought about doing sort of like next level to just keep that extension going and just or have you looked at this or thought of looking at this so that the NOD mice sort of in question never get type 1? Is that a possibility?
Yeah, that's definitely something that we've thought about. Honestly, we're waiting a little bit on some more NIH funding to come in, just because it's been a little bit backlogged lately. But that's definitely something we're looking into. If we could continuously dose, how long could we go without seeing onset? And what would that look like in terms of the various immune cell populations that would result? Could we kind of understand some flow cytometry markers, or even some sort of markers within the blood that would determine, oh, when could you stop? That's one of the questions we have, especially with the primate studies, is what are the biomarkers to tell you that you no longer need to dose and, the tolerance would be sustained. So that's something we're looking into. I'm sure a lot of people are looking into that as well. So excited to see more.
Hopefully there'll be some cross communications with people's findings. And then I guess on the flip side would be, what would be, you'd have to also be alerted to the biomarkers that show you're sort of coming out of remission as well, right?
Yeah. I think that could be indicated by your autoantibody titers, but there might be some other more like earlier markers that could tell you that as well.
You wonder if you could even use a CGM inflections or,
yes.
Postprandial CGM readings to kind of suggest that things were, that there was time for a redose. So.
Yeah. A1C or C-peptide even. So yeah.
And then in terms of the subcutaneous administration, did you note any adverse effects in the places that it was administered in the body?
No, we didn't see any adverse effects for the tolerazome group. We had a control group that was a subcutaneous rapamycin, and we did see alopecia or hair stop growing at the site of injection for the control rapamycin group, but that was not the case with the tolerazome group. Obviously, side effects are different in mice and primates and humans, so there'll be other things we need to look out for as we kind of scale forward.
Yeah, you just wonder like, okay, is it, are there better injection sites subcutaneously versus others? That's what I was kind of getting at. Here's a question from Kiati Gerder. What is the effect of these nanoparticles on APCs? She might have joined a little bit later. And does it delay the onset in male nod too? What does absorbance look like in different organs?
Yeah, so it reduces the co-receptor expression in antigen-presenting cells, so dendritic cells, monocyte and macrophage-like cells. And then the question, we have not gone for the male nods yet, but that's definitely something to consider with gender differences. And we would also like to do some adaptive transfer studies with the male mouse as well, just because they don't onset as well, to see what we can understand from that result. And then I think you're asking about biodistribution for the last question. So when you give the particles subcutaneously, they do drain well to the lymph nodes, the draining lymph nodes, depending on the site, like you're saying. So we give them in the next graph in the mouse, they drain to the axial and brachial lymph nodes. And so, placement of the subcutaneous injection would be something to consider, especially for humans. We do find that you get some of the particles to the inguinal lymph nodes, so they are going throughout the lymphatic circulation into the other APC populations within the body. And then all nanoparticles have some clearance to the liver. It's less of an issue when you give subcutaneous than intravenous. If you do give IV for these particles, you get more particle going to the spleen. So if you did want to target the spleen for any reason, the IV administration is good in that regard.
Thank you for answering those questions. And then I just had a question for you. This deliverable mechanism seems like it might be useful in other autoimmune diseases. What are your thoughts on that? Is it just sort of one and done for type 1, or are there other opportunities for this type of delivery system?
So I think that we rely on the presentation of the antigen, right? And so if you have something where we can define the onset very, very specifically, and we're able to do that now with the staging kind of protocol for type 1, that's something that I think this could be applied to right now. Like something like lupus is very hard because sometimes people aren't diagnosed very quickly. They might be diagnosed over 1 or 2 years. So if we have this antigen presentation and the treatment schedule that can be aligned together, that would be really appropriate. I'm not sure it would be more of a logistical challenge to align things than sort of a scientific or medical challenge with other diseases.
Yeah, it does. I think it really speaks directly to the fact that the screening and the understanding where people are in the course of their march towards type 1 is going to be so important therapeutically. So thank you so much, Jacqueline. This is a really exciting approach, and we will be watching to see as you progress to your next steps. Really hope, fingers crossed that everything moves forward for you in the regulatory space, and appreciate your taking the time to speak with us today.
Thank you so much. Have a great day.